Your cell's sugar coat controls the spark inside your brain. Everything we thought about how neurons communicate might be missing a vital piece of information. Think of the plasma membrane like an incredibly sensitive electronic filter. Normally, we just think this barrier separates things, but it's also dynamically tuned by the elaborate sugars covering its surface. These molecules aren't passive decorations. They are microscopic topological structures that literally decide which electrical signals get through and how fast they fire. The specific arrangement of these glycans modulates local ion flux, physically dictating your cognitive state. It is a profound interplay. The subtle architecture determines global awareness. The research shows this mechanism goes far beyond simple docking receptors. It suggests the membrane itself acts like an informational boundary, a filter tuned by sugar architecture. Imagine the surface charge of your cell being micro-adjusted. These subtle glycosylation patterns physically alter local hydration shells, changing the statistical probability for ion channels to open or close in immediate proximity. But it's not just about static electricity. When those glycan chains restructure how lipids interact, they change membrane curvature itself. This topological shift governs fluidity, a biological rheostat that directly tunes the dynamics of coherent neural firing across large populations of cells. It is a mechanism where nanoscale structure dictates macroscale coherence. Bridging biochemistry to awareness has never been more precise.
But this leads us to a staggering bottleneck in modern neuroscience. We have observed that these nanoscopic changes, these sugar coat shifts altering membrane dynamics, are happening constantly, yet we possess absolutely zero way to non-invasively map them onto subjective experience itself. How do you measure deep awareness or the state of flow? The current technology cannot translate a subtle alteration in glycoform topology into an observable shift in global brain coherence. It is like trying to feel music with only static electricity. This gap between microscopic structure and macroscale consciousness is where the revolution lies. What if we could build detectors sensitive enough not just to see proteins, but to read the language of these living sugar architectures, which means something nobody expected for how profoundly linked structural biology truly is to our felt reality.
This structural role elevates the plasma membrane from a simple gatekeeper to an active conductor of internal state. Think of it as every cell possessing its own hyper-fine tuning mechanism for reality itself. The specific glycosylation patterns are not merely informational flags. They act like microscopic frequency regulators within the synapse, literally setting the bandwidth through which information travels and is interpreted. This glycosynaptic interface suggests that consciousness isn't just an electrical discharge, it's a precise physical architecture being constantly orchestrated by sugar motifs. It means deep awareness might be predictable based on these patterns alone. Yet, if this tuning mechanism determines subjective states so fundamentally, how universal can it possibly be? Some researchers suggest entirely different metabolic pathways govern neuronal stability under high stress. Who is right about the fundamental rules of biological signal coherence? The ultimate insight here is the transition from cellular mechanism to complex system behavior. What we are mapping isn't just a local charge change, it's how those precise sugar motifs dictate global coherence across vast neuronal networks, treating the brain not as circuits, but as a self-organizing topological entity. The plasma membrane ceases being an individual barrier and becomes part of a massive adaptive architecture, a system whose patterns define its function. This is where physics meets phenomenology. Structure determines awareness itself. Shifts in glycan typology aren't small biological noise. They are the fundamental coordinates for shifting into states like deep flow or acute focus. The brain state isn't just driven by neurotransmitter balance. It is dynamically sculpted moment to moment by its living sugar coat. If we can quantify this architectural influence, does that give us control over the very parameters of subjective experience? But what if those patterns aren't just tuning devices? What if they are physical architects? Consider this. When specific lectin molecules bind to a sugar cluster on your membrane, it doesn't gently nudge a receptor open. That binding physically restructures the domain itself. It reorganizes protein architecture by brute force at the nanoscale interface. This means we're talking about highly deterministic cellular signaling pathways.
The sugars aren't just modulating flow, they are deciding which entire signal path is allowed to exist and where it terminates. The implications shift from subtle influence to physical control over complex information transduction. If this deep level of structural plasticity dictates these high-level outcomes like awareness or cognitive collapse, then we need instrumentation that can watch the scaffolding itself reassemble in real time. But even if we capture that instant, what does watching a cellular domain rearrange really tell us about intent?
Here is where everything changes. We have been viewing the cell membrane as merely modulating electrical signals, like a biological valve opening or closing based on its sugar coat. That interpretation is too simplistic. It misses the fundamental physics at play. The glycan architecture doesn't just tune the channels, it actively governs energetic channeling itself. Think of local memory structures not as storage banks, but as physical regulators for energy flow across the membrane's curvature. This biophotonic flow regulation suggests that our conscious states are tied to coherent streams of informational current running through the cellular structure, a dynamic, measurable field dictated by topography. It implies subjective awareness isn't merely a chemical reaction, it is an emergent phenomenon driven by these energetic currents. We have been looking at biology as mechanics when we should be observing it as physics in motion. If consciousness can truly manifest as such coherent energy flow guided by molecular shape, what does that mean for the boundary between life and information?
The profound connection we've traced suggests that our capacity for states like perfect concentration or deep intuitive understanding might not be a matter of willpower alone. It could be an engineered physical outcome, dictated by the optimal topological geometry of cellular surfaces across your entire nervous system. Think of consciousness as an infinitely variable antenna array, and these sugar motifs are the precision alignment controls, tuning in specific cosmic signals, specific cognitive states. If we can learn to shift those glycoforms intentionally, not through chemistry alone, but perhaps through highly focused somatic or environmental cues, then awareness becomes a navigable dimension rather than just a passive experience. We move from simply observing how cells operate to understanding that we may be able to consciously sculpt the physical architecture underlying our own perception. This is the boundary between biology and engineering itself. We started by asking how our brain sparks. We end having mapped the physical language of awareness itself. The complexity reveals an elegant truth. Your cellular architecture holds the key to tuning subjective state, flow, focus, intuition, with precision that was previously considered purely philosophical. Your actionable takeaway is this: when you feel a shift in deep concentration or unexpected clarity, do not dismiss it as luck. Instead, recognize it immediately as evidence of a topological refinement within your own nervous system's signal architecture. Treat the state itself as observable data. This understanding changes everything about how we define mind and matter. The era of passive biology is over. This knowledge provides agency in our inner lives. The full research archive is at jmthomasofficial.com.